this live session. So I hope you have been having a nice time learning about biostatistics. I presume from the responses and the doubts which have been raised in the online forum that a lot of you are more comfortable during the first half of the course and lot more queries for the second half of a course where you start to apply concepts of probability, central limit theorem to practical problems. So I had a sheet shared with me for some of the queries which were raised. So before I address the queries which are raised right now, so I will first read out the queries listed by students in the Excel sheet from before. So one of the first questions is by Likhit Sai Funny. It's not clear to me it's a he or a she, but the question is uses of biostatistics for biotechnology. So one thing you must realize is that we as a student, we tend to segregate different fields. In reality, everything is connected. So as a biologist, you routinely do, for example, protein estimation. If you want to do a Western blotting, you need to make sure that between two samples, you load proteins of equal amount. Now how do you know you are loading proteins of equal amount? As part of that, you run a BSA standard and determine the protein concentration of the sample that you have prepared. So what is the process there? You are essentially making use of linear regression. So there you make solutions of different concentrations of BSA, you generate a standard curve, and then so you generate a curve between OD and concentration. So for known concentration of BSA, what is the OD value that you record? That way you fit a line, and that is your standard curve. Now when you have an unknown sample for which the protein concentration is not known, what you do is you first take the OD reading, and then making use of the line fit which you obtain based on experimentally observed data, you backtrack and try to figure out what the protein concentration is. So this is a very standard thing which you do. Probably you have been doing it unknowingly, that you are making use of concepts of linear regression for determining protein concentration estimate. Another example. So you want to use a drug, you have developed a drug perhaps which inhibits proliferation or inhibits migration in the context of cancer. So you want to figure out what is the concentration where you would significantly inhibit migration by 80%. How do you arrive at that number? What drug concentration to use? For these purposes, what you do, so you tweak the concentration. You have multiple concentrations from 0 micromolar, let's say, to 50 micromolar. And for each of these concentrations, experimentally what you have measured is, let's say, cell motility or cell proliferation rate from MTT. Now you get a curve. How do you figure out where is 80% of motility is impeded? One is simply to do the data fitting with a particular curve. With the curve might look exponential, or the curve might be a sigmoidal in nature, that for a certain concentration your motility is unchanged, and then as you increase the concentration you see drop in motility. So here also you are making use concepts of data fitting. Now let's say another case, you have a drug which you want to make use and which should be effective in hypoxic environments in the tumor. So you can have a normoxic and a hypoxic condition, and in these two conditions you want to see what is the concentration of drug which is effective at both the conditions. So here you have two variables. One is your drug concentration, and the other one is your condition, which is normoxia or hypoxia. So here you will make use of two-way ANOVA to figure out what concentration of drug you would require so that it is effective at both conditions. So all these are prototypical examples where in essence the problem is a biological question, but you have quantitative data which you are making where you are making use of concepts from biostatistics to answer the question. So the second question was raised by Piyusha. She says, which book do you recommend for statistics in data science for biology? So I don't exactly know what you mean by data science for biology. Is it just statistics, or data science you mean machine learning, artificial intelligence? I am not aware of that right away, but you can just for this particular course, for clearing your doubts pertaining to statistics, you can look up the book by Introduction to Probability and Statistics by Meadenhall, Beaver. This is by Cengage Learning. I think the book is priced around 800 bucks. You might be able to get a cheaper version of that online as well. The next question is raised by Padmaja. Can you please suggest methods in which the students can approach studying for the exam? So the easiest method is to put more time, is to try and solve problems by oneself instead of just looking up problems which are already been solved. So you might, let's say the book that I referred to, you can download, you can buy a copy of that book, go chapter by chapter, and at the end of the chapter, whatever questions there are, you attempt at least 10 to 15 of the list of problems. That is the only way in which a concept will be clear to you over time. The next question, Dhimant Sheth has asked, all statistical calculation applicable for when data is having normal distribution. How to check distribution of data? Part 1. And the second part of the question is, in any case, if data does not follow normal distribution, then what is the way forward to it? Okay, so this is a very important question. I'll answer the first part of the question, which is how to check distribution of data, or whether, so in statistics, so most of the techniques which have been taught in this course assume that data is sampled from a normally distributed population, so on and so forth. So all data which is drawn from a normal distribution is called parametric data. Okay, and most of the techniques that were taught in this course were parametric tests. However, there are data which are non-normal in nature. Now, how do you figure out whether a data set is pulled from a normal distribution? So one easy way of doing it is, if you have a set of data and you want to know whether that is drawn from a normal distribution, is to see whether for the data that you have collected, does it follow the principles or the different aspects of a normal distribution. For example, for a normal distribution, you know that 65% of the data should lie between mean plus minus 1 standard deviation, 95% between mu plus minus 2 standard deviation, and 99% between mu plus minus 3 standard deviation. So what you can do is, from the data that you have collected, you can find out the value of mean standard deviation, and then you convert these raw scores into a z-score. So you will do x minus your mean that you have obtained divided by standard deviation, that is your z-score. And so for z-score, you know that between each point, so your z-score between -1 and +1, you should have 65% of the data which should fall within that pool when you draw the histogram for the z-score. So this is a simple way of doing it. Another property of your normal distribution is that it is a symmetric distribution. So you can, let's say, you calculate the value of skewness or kurtosis. So kurtosis for a normal distribution, the value is 3. So if you calculate the skewness, if it lies between plus minus 0.5, then you can reasonably assume that the distribution is kind of symmetric. If your kurtosis is more than 2.5, you can be reasonably assured that this follows a normal distribution. However, there are more stringent tests. For example, there are other tests to check the normality of the data. One such test is Kolmogorov test. Another one is I forget the name, I will send that email, but there are multiple other tests to check the normality of the data. Now, okay, so if now you have done your normality test, your data is non-parametric, what kind of statistical test do you use? So equivalent of your t-test, you have something called Wilcoxon sum rank test. This is for non-parametric data. Similarly, equivalent of your F test, that you in one way ANOVA, two ANOVA, you do that F test. You have a Kruskal-Wallis H test. This is doing the same thing but for non-parametric data. Now interestingly, you can make use of non-parametric tests even for your parametric data as well. Okay, so there are some scientists who believe that the non-parametric tests are equal if not more robust than many of the parametric tests. So because these are more advanced, and so these were not covered as part of the course, but you are welcome to look it up. So there is the next question by Bigneshwar. He asks how to know where to use sigma root n or sigma by root n. Okay, so this pertains to central limit theorem. So as per central limit theorem, if you have random variables x1, x2, x3 which are independently drawn from a distribution with mean mu and standard deviation sigma, then the mean of these random variables, so x bar, if you define x bar as x1 plus x2 dot dot xn, that will follow normal distribution with mean mu, which is the mean of the population, and variance sigma square by n, which means the standard deviation will be sigma by root n. So if you are comparing sample means, then you will use sigma by root n. However, if you are concerned about sum of the variables x1, x2, x3, in that case the variance, because x1, x2 each of them are independent of each other, variance of summation of xi is simply summation of variance of xi. Since each variance is sigma square, so you have net variance of summation of xy is sigma square n. In that case the standard deviation will be sigma root n. Okay, so let us say we have a problem like for an insurance company, there are multiple people who have bought insurance. Okay, in that case x1, x2 refer to the insurance policy of each agent, of each individual. Okay, now when we talk about the insurance company itself, it wants to calculate how much, let's say, annual charge it will charge, so that it makes sure that it does not make a loss, or there is less than 1% or 0.001% chance of making a loss. Then in those cases you will make use of summation of xy. Okay, so the next question, Dr. Nalini Kurup has added, in week 5 and 6 lectures there were so many formula derivations, but when I did week 6 assignments I found it difficult to apply the right formulas. Can you please give a list of formulas and how to apply for a particular question? So what I will do is I will upload a document in the NPTEL website with some prototypical examples, but once again I would like to reiterate the best way of clarifying your doubts is by actually attempting questions at the end of a chapter. So you pick up any biostatistics book which covers this syllabus, you go and solve the problems, then you will know which formula is applicable when. Okay, the next question is by Ananya. Many solutions to the assignments are seeming doubtful, so can the stepwise solutions to the sums be made available? So if I am not mistaken, my TA Vivida has already uploaded the stepwise solutions for week 1 to 4 assignments. Since 7 weeks are already over, I will ask my other TA Asad to upload the stepwise solutions to assignments from 5 to 7, and once the last week of classes are over, then he will also upload the last week's solutions. Okay, now let's come to the next question. Biostatistics, what type of statistical tools used? Raised by Gangu Varalakshmi. So your question is not clear to me at all. What do you mean by what type of statistical tools used? You can use, so whatever we have taught throughout the course, like one-way ANOVA, finding out mean, doing linear regression. The very first example that I discussed was where you are using different aspects of biostatistics to get some information which is biologically relevant. Okay, beyond that your question is too vague for me to clearly answer what exactly you want to know. Okay, so Shraddha Gupta has asked what is the detailed format of final exam. So if you know already that as per NPTEL has a like set format of questions, you'll be asked multiple choice questions from my lecture, the assignment, and the last section will be open questions where you will have multiple choice questions like just like the other sections, maybe a little bit more difficult, and there will be some fill in the blanks type of question which require you to solve the question and write down the answer. Okay, so that brings me to the end of the questions that have been raised thus far. I will wait for some more time and then we can take it from there. Okay, so is Pearson okay, so there is one question asked by Mrinalini Singh. Is Pearson correlation robust in case the assumption of normality is violated? Pearson, for calculating correlation coefficient, there is no assumption of the type of the data, but so at least with correlation coefficient there is no difficulty so far as whether the data is normal or non-normal. What is more important there, if you are doing linear regression, then whether the data is linear or not, that is the more important point as to what kind of correlation you will draw. But the correlation coefficient metric in itself is robust. Okay. Can we use biostats to analyze qualitative data? So it again boils down, so what you are raising is analyzing non-parametric data. For example, let's say if we are comparing between the quality of food offered by 5 different restaurants, and there are 10 subjects who are individually assessing each of them. So this is an example of a qualitative data. You will say you rate your response between something like 1 and 5, 5 being very good, 1 is very bad. In these cases, so what is important is the rank. Okay, so you can, so that's what I said for non-parametric data. Wilcoxon sum rank text is a sample example of how you make use of something for non-parametric data and which is equivalent of your t-test. So how to find probability of proportion? There is a similar approach. So in this book of Medenhall, Beaver, you have some description of how to do proportion. I request you to look it up. Using Excel, MS Excel, how component bar diagram is drawn? I don't understand what you mean by component bar diagram. Is it simple bar diagram or is it something different? I don't know exactly what you mean by component bar diagram. Can you be a little bit more specific? So Ananya Mukhopadhyay asks, where is the link of the solutions? Couldn't find it. Can you share the link? So I will ask my TA Vividha to repost the link of stepwise solutions for weeks 1 to 4. So Vigneshwar asked this question for microbial MIC study. Which stats test is more suitable? For insecticidal study, is probit analysis good to interpret lethal concentration, or any other method is more suitable? So my honest answer is I don't know what is probit analysis. I'll ask my PS, do you know what is probit analysis? Okay, so I am not aware of it. I will, but to find out the minimum inhibitory concentration for any particular case, you would fit your data to a sigma model and find out what is the 50% drop in viability. So that can be done mathematically using fitting of nonlinear functions. Lecture 28, average weight between 167 and standard deviation 27 in a population. Sample of 36. What is the probability that average weight is between 163 and 170? Okay, so what it is saying that you are given the mean of the population, which is 167 and standard deviation is 27. Okay, so this is mu and sigma of the population. So now you have to calculate, if your sample is 36, what is the probability that the average weight is between 163 and 170? So this should be, I don't want to give you the answer, then it is as good as that I am telling you what to do, but you figure out. So what is the probability that if x is the random variable which is your weight, what is the probability that x lies between 163 and 170? So how can you make use of the central limit theorem to do this? All the information you have been given, let me tell you, you know mu, you know sigma, you are given small n. Now you can do from here. Okay, Manish, I tried to draw a bar diagram through MS Excel. One of the type of bar diagrams which I study in is component bar diagram, in which all the values are expressed in single bar by splitting it. Oh, okay. So I probably understand what you are saying. So let's say if we have this, I could draw manually. I think it is like, let's say, if you want to know, so you have cells which are in their different stages of cell cycle, let's say S1 phase, G phase, M phase, but the total population is 100%, and then you need to know, so between this and let's say in another case where you perturb with one drug, you want to know what is the relative distribution of the individual cell cycle stages in the whole population. So the population is 100%, and how to do, so the individual fractions within that 100% will get divided. So off the back of my mind, I don't know how to plot it in Excel. I will look it up and I will post that information on the online forum. So Asad, can you please jot down? Okay, so how instructions for drawing a component bar diagram. This information will be shared with you, so we will make a short document of the steps that one needs to make in order to do this kind of plot. I probably think you have to make 4 columns. Most likely you have to make 4 columns. You choose the column together and then you say draw a component bar diagram. That should do, but still I will check and let you know. Vignesh asks, sir, please explain the W6Q noun. I don't understand what is W6Q9. It's W6Q9. I don't know what you are referring to.
Week 6 question number 9.
Okay, week 6 question number 9. Do we have that information with us?
We have the question. Yeah, we have the question.
Okay, what is it?
So actually here we have to use. So this is not in the course. So he tell him that he has to read extra. So I had explained.
So my TA Asad tells that this was not covered in the course and you are instructed to read some extra material. Okay. So Nancy asks box plot and upper fence value. So in a box plot there are 5 lines. Okay. One is your Q1. So the box itself has Q1, which is the 25th percentile, the median, and Q3, which is the 75th percentile, and the 2 lines which stick out from it go to the lowest and the highest point, or though they actually list the points within which, given a data point, you will not consider as an outlier. So any point which lies beyond those 2 fences are outliers. So the lower value is basically Q1 minus 1.5 times interquartile range, which is Q3 minus Q1. Okay. Manish Kumar, one more question: how to draw a box plot and what are the outliers in the box plot? So I just explained. So once again, so the box itself, the lower line is your Q1 or the 25th percentile, the upper line is Q3 or the 75th percentile, the middle line is the 50th percentile, which is same as the median. You have another separate point in the center which shows you the position of the mean. So if you have skewed data, your median and mean may not lie on the same point. Now there are 2 lines which extend out of the box. The lowest point till the box extends is the position Q1 minus 1.5 times interquartile range, which is Q3 minus Q1. So if you have any point which is lower than that point, you can consider that point as an outlier. Similarly, the topmost line is basically the position demarcates the line Q3 plus 1.5 times interquartile range. So any point above that is also an outlier. So how to draw box plot? How to draw box plot? So at least in softwares like Origin, you have the option of choosing the box plot for your data. I am not sure whether in Excel also you have that option. Most likely in Excel you cannot plot the box plot by itself, but statistical softwares like SPSS, Origin, anything else will give you that option. Probably you can plot a box plot in MATLAB as well using functions. Okay. Parinita asks whether such out of the course questions will be asked in exam. Yes, the third portion of the exam will be on questions which I frame to test your understanding of the subject. But the syllabus will be from whatever you learn through the course. Okay, so there is a question from Nancy. Sir, please explain percentile calculation. So the pth percentile, okay, so the position, the pth percentile you calculate as p by 100 into n plus 1, where n is the total number of data points. Okay, so that would mean that if you are talking about the median, that p is equal to 50. So you have 50 by 100, which is 0.5 into n plus 1. This is the position, it is not a value, this is the position at which you calculate. Now let's just hypothetically say for a given data set that n plus 1 by 2 becomes, so if you have 5 numbers, a data set of 5 numbers, the third number is your median. But let's say for a data set of 6 numbers, so 50% of n plus 1, 6 plus 1, 7, so 7 by 2 is 3.5. In that case, you are doing the averaging. So similarly, let's say you are calculating the 20th percentile for a data set of 6 numbers. So what is the position of the 20th percentile? The 20th percentile position is 0.2 into 6 plus 1. So position is 1.4. That is the position of the percentile. Now how do you calculate that number? So that number is the number at position 1 plus 0.4 times the difference between the number at position 2 and the number at position 1. So there is a question from Kadir. The course introduction to is it exclusively for engineering students or for medical students as well? It is supposed to be for all students. However, the course does require you to invest significant amount of time to make yourself conversant with the data. So while some of the concepts taught in the course you might find a little bit difficult, but lot of it, at least at the assignment level, most of it is very straightforward if you are following the course very carefully. So Ananya has asked this question: which test can be used to draw an inference about the ill effects of social networkings in adolescents while doing a study? So I presume that the type of data you are talking about is non-parametric in nature. So you will have a way of somehow explaining something which is qualitative in terms of numbers, so will still not be absolute numeric data. So this is non-parametric data. You can do the Wilcoxon sum-ram test or the Kruskal-Wallis H test. If you want to do something equivalent of ANOVA, then Kruskal-Wallis is the test to do. Okay, Nancy asks, can we use white paper and pen for the exam for doing calculations? So if I am not mistaken, the terminals will have inbuilt calculators which you can use, and white paper will be provided at all the exam centers. It's not that you have to do all the calculations in your mind. Okay, so Manish Kumar asked this question: sir, what should be minimum number of items values in the sample when statistical methods tools can be applied? Of course, in an ideal world you will need as many samples as you want, but t-test is specifically designed to handle when your sample size is very small. So if you have only 2 values, then of course doesn't make any sense to do any kind of statistical test. But if you have even 5 pieces of data, then you can make use of it. T-test for comparison or just mean median can always be done. Something jotted down.
What I wrote that one question.
Few examples of stepwise how to make the formula, and that what type of plot? Companion bar. These two we should upload. Okay. Manish Kumar, I mean for applying standard deviation. So, of course, your standard deviation, if you have very few number of observations, your standard deviation is going to be high. Okay, so that goes, or you might even have a low standard deviation depending on what data you obtained, but that is the very reason when you are reporting standard deviation, you should have at least 5 or 6 values for to make that calculation. So Vigneshwar asks when to use ANOVA but not t-test. So see, ANOVA, when you have multiple samples, let's say think of the case of you're using, you're treating cells with drugs at varying different concentrations. So there are two aspects to ask. One is the question, is the drug doing something to the cells? Number one. And second is at what concentration? So if you have the whole set of data, then ANOVA will first, when you apply, do one. This is an example of one-way ANOVA. When you are doing ANOVA, you will first get the answer that yes, the drug has an effect, but that is not full, that is not complete information, because you still don't know at what concentration is the drug having an effect. So the next stage, what you do is a post hoc test, where you do a pairwise comparison and see whether any two values, the difference is statistically significant or not. Now you can do theoretically t-test 2 at a time, but because there is always a possibility of error, right? Alpha is that error where we reject H0 when it is true. So if you do multiple t-tests, then the probability of making an error increases. ANOVA is that technique which allows you to compare across the conditions first to reach that conclusion, yes, this drug is working or not. Then if, let's say, if the drug does not have any effect, then you stop there. There is no question of then comparing any two values and saying this is significant or not. So if, let's say, you have 5 different concentrations of drugs, and even in one case, let's say, the drug has an effect, your ANOVA will tell that yes, the drug is effective. Then you do pairwise comparison to figure out which one is, between which two points there is statistical significance or not. Okay. Kadir asks ideal sample size for randomized controlled trial. So this is where, when you are doing sampling, ultimately you want to get information about a population. And why is the central limit theorem so powerful? Because it says that for sample size, irrespective of what is the distribution of the population, if you take sample size as little as 31, then you are guaranteed that the sampling distribution will be normally distributed. Okay, so that's why choosing a sample size greater than 31, you are bound to make use of parametric data sets. Okay. So in that case, but let's say you have a situation of where you cannot have those many number of samples. In that case, you have to rely, you are taking smaller sample size, but then you make use of t-test for doing comparisons. Okay. Please explain, Nancy asks, please explain Z alpha and Z alpha by 2. Okay. So between two conditions, let's say I want to compare between two populations, and in one case I want to make a statement between the two populations are similar, or in other words, my H0 is mu1 equal to mu2, right? So I don't care, my alternate hypothesis is mu1 not equal to mu2. So you can have mu1 greater than mu2 or mu1 less than mu2. So in that case, you would make use of Z alpha by 2. This is a two-tailed test. But if in a setup I want to ask the question whether mu1 is greater than mu2, so in that case you do a one-tailed test and you make use of Z alpha. Okay, so Manish has asked, my question has practical application, like in my university when CGPA is calculated, standard deviation is applied on marks of those classes where number of students is more than 30. Okay, so standard deviation is when CGPA is calculated. Standard deviation is applied when CGPA is calculated. Standard deviation is applied on marks of those classes where number of student is more than. I don't understand the connection between calculating standard deviation when marks of students is more than, when number of students is more than 30. Even between 10 data points I can calculate a standard deviation. So there is nothing to do with, so the error associated with estimating the standard deviation will increase if your sample size is very small. That is the only thing that one needs to think of. So in the Excel sheet, Ranjana has asked a question, how to find out sample size from mean and standard deviation. So this question is not properly posed because so let's say you have a situation where the mean, you get some value of the mean and the standard deviation is very less. Now that might be because the sample size is very large, or inherently there is very little difference between the individual data points. So you cannot directly connect between mean and standard deviation with sample size. So Vigneshwar asks another question, additional question, how to select which post-hoc analysis like Tukey or Bonferroni or any other. You can stick to any one of them. Each of these post-hoc tests have their advantage and disadvantage. The safest thing is to use 2 or 3 of them, and if all of them are consistently telling you that a difference exists, then you can be, your measurement, your quantification is robust. So to answer your question, Nancy, this is just the same question which Vigneshwar asked. How will we assume which post-hoc test is suitable after ANOVA? You can do Tukey's test, you can do Bonferroni, you can even do t-test. There is nothing wrong about it. So long as, so if you read a paper, you will see typically in materials and methods there is a section called statistical analysis. The authors clearly state what post-hoc tests they have used. So any of these tests you can use for parametric data. You use Mann-Whitney U-test is an example of a post-hoc test used for non-parametric data. Okay, Manisha's histogram is prepared and marks are moderated in classes having strength more than 30. This changes the actual SGPA of the students. I think that that is done just to make sure that you have a bell-shaped distribution for the entire class. So I will leave in 5 more minutes. If you have some more questions, please ask. Otherwise, you are welcome to post your message on the online forum. We will collect those queries and again post an updated document with answers to all those doubts. So the choice of 30 Manish might have to do with application of central limit theorem. Kadir asks, if I am studying the efficacy of a particular drug on sample particular drug on sample of 50, then at the end of the study, what statistical technique I can use for final conclusion? So if your sample size is 50, okay, and you are in case of drug, let's say you are doing your data is parametric in nature in terms of viability, survival, whatever. Okay, so you can use ANOVA if you have multiple drug concentrations. If you are using only one particular concentration for the drug, you can use normal distribution, simple comparison, t-test also. So as all of you would recall that in t-test table you have degree of freedom till 29. After 29, n equal to 29, you see that the t-test, the t distribution and the normal distribution completely overlap on each other. So for any size greater than 29, this is the normal distribution that you can apply. So as per central limit theorem, your sample mean will follow normal distribution with mean equal to same as population mean, and sigma is equal to sigma by root n, not sigma, s equal to sigma by root n. डाउट्स भी और कुछ नहीं है.
पोस्ट ऑन ऑनलाइन if you get any question.
हाँ
Post on online forum.
हाँ
Any question.
हाँ ठीक. Okay, so if you have any further doubts, I welcome you to raise these queries on the online forum. Vividha and Asad will collate those questions, will prepare the answers and will repost on online. And I'd like to wish you best of luck for the final exams. Please prepare well. So MCQs are reasonably easy, the other questions little bit tricky, but if you know your stuff, it will be a cakewalk. Thank you.